Hello. Today we will be going over a widely used scientific technique known as CRISPR. Presenting this system will be
Alonso Treviño,
Elise Gutierrez,
and Stephanie Cavazos.
Clustered, regularly short palindromic repeats, better known as CRISPR, is a method that takes into consideration the very explicit and quick adjustment of DNA in a genome. This system is what gives a total arrangement of genetic instructions in an organism. There were three major findings of CRISPR that will be mentioned in this video. The first discovery of CRISPR goes back to 1987 when a Japanese scientist from Osaka University named Yoshizumi Ishino, along with a group of co-workers, accidentally identified CRISPR on E. coli bacteria by using the isozyme conversion of alkaline phosphatase, also known as the IAP gene. They discovered that CRISPR can do genetic modifications along with the enzyme Cas9, as well as noticing that the gene segments of DNA had 5 short repeating segments, which were separated by a short non-repeating spacer. This spacer had its own and unique sequence, which was composed of 32 bases, compared to the sequence of the repeating segments, which had identical sequences and were composed of 29 bases. It was the first time that the CRISPR mechanism was published. Also, the repeats found on a DNA sequence are palindromic, which defines as a particular area of a DNA strand in which the grouping or sequence of nucleotides is exactly the same with a reverse sequence on the complementary strand, reading the same left to right. They later continued with their research, and in 1990, they had started to find, with the guide of new enhancements to DNA sequencing, that this example was pervasive in a wide range of microorganism species. Then, in 1993, Spanish microbiologist Francisco Mojica found out that the DNA of a bacteriophage, which is a virus that lives inside a bacteria and is capable of replicating as well as destroying a bacterial cell. This goes hand in hand with the functions of CRISPR, which can be used to fight bacterium in an altered immune system. He discovered that spacers found in DNA matched those found on bacteriophages. Lastly, in 2005, Alexander Balatin, who was also a microbiologist, was the first to hypothesize how CRISPR gave resistance, suggesting that transcripts from the CRISPR locus worked against sense RNA inhibition of phage gene expression. His supposition would end up being incorrectly.
CRISPR is an influential system that can be extremely useful in the cell biology laboratory. The scientists knew they could utilize CRISPR sequences of DNA as well as guide RNA to locate target DNA and then arrange CRISPR-associated enzymes as molecular scissors. As we know, scissors serve to cut, modify, and in this case, replace genetic material. This discovery was amazing because it opened a world of possibilities in the engineering of human DNA. What is the takeaway from all this? The takeaway is that scientists had a precise and quick system to alter, delete, and rearrange the DNA in almost any living organism. Before, they would utilize genome engineering methods like zinc finger nucleases or transcription activator like effector nucleases, which required them to design and create new nuclease pairs for every genome target. CRISPR is a simple and adaptable system which now makes it the most widely used genome engineering approach. As mentioned before, CRISPR has two components, a guide RNA and a CRISPR-associated endonuclease, also known as Cas protein. The gene RNA serves as a guide for the insertion or deletion of uridine residues into the mitochondrial mRNAs. The target sequence in gene RNA is important in changing genomic target of the Cas protein. The original discovery led scientists to use CRISPR for knocking out target genes in cells and organisms, but with modification to certain enzymes. CRISPR has widened the scope of use in selectively activating and repressing target genes, purifying specific regions of DNA, and precisely editing DNA, and RNA. CRISPR is the most popular system used when scientists want to edit the genome of an organism. When using CRISPR in your cell biology laboratory, there are different manipulations to consider. Do you want to generate complete and permanent loss of gene expression or function, which is also known as knockout? Or do you want to generate a specific mutant allele of a gene, also known as point mutant? Or do you want to increase or decrease expression of a target gene? In the process of CRISPR, we begin with Cas9, which is an RNA-guided protein that goes on and searches the genetic material trying to detect DNA sequences. For example, a sequence of A's, T's, G's, and C's that are compatible to the sequence in the guide RNA, which have already been programmed. The guide RNA then guides the Cas9 to open the DNA helix and then positions the molecular scissors to cut each strand of the DNA's double helix, causing a double strand break. At this point, the repair enzymes are triggered in this unnatural event that was induced by the Cas9. However, the enzymes are ready to seal the gap of the fragmented DNA. Sometimes they insert new genetic information garnered by the cell or the scientist. However, the cell is unaware of the situation that took place, meaning it doesn't know whether the DNA was removed or added. So it basically goes into the cell and repairs it blindly. This causes the insertion or deletion, also known as indel. It is important to note that the mutation is not caused by the cutting of the DNA, but from the actual insertion or deletion that occurs in the process. This system allows for the correction of disease-causing mutations by changing the original genetic code.
Once scientists began to understand how CRISPR functions in bacteria, they realized that CRISPR could be used as a powerful tool for genetic engineering and editing. By working as molecular scissors, CRISPR essentially cuts out unwanted sequences and replaces them with a new one. Scientists have found a way to use CRISPR on any organism, including humans. Therefore, CRISPR can cut out genes that cause serious genetic illnesses, such as sickle cell anemia and cystic fibrosis. They can then replace those dysfunctional genes with the correct functional genes. Thus, CRISPR can be the key to curing these diseases and many others by using so-called gene therapy. Furthermore, scientists have been exploring the possibilities of using CRISPR to better our agriculture. In agriculture, CRISPR can be used to make plants resistant to harmful insects and diseases, or it can be used to yield more or larger fruits and vegetables. The applications of CRISPR seem endless. Scientists, however, are still in the beginning stages of experimentation and are testing it endlessly to make sure CRISPR is safe in the long run.